Welcome back to the afternoon, and I'd like to introduce this afternoon's keynote speaker, Susan Lindquist. Sue has been a member of Whitehead Institute for 10 years and is my predecessor as director of the Institute. Those of you who attended last year's colloquium may recall that at that time we had the pleasure of celebrating Sue's receipt of the 2010 National Medal of Science. She was recognized by President Obama for her studies of protein folding, which are having profound implications for fields as wide-ranging, and this is the quote, for fields as wide-ranging as human disease, evolution, and biomaterials. In addition to being recognized with our nation's highest scientific honor, in 2010, Sue was also awarded the Mendel Medal from the Genetic Society in the United Kingdom, and in addition, the Max Delbruck Medal. These latest awards added to Sue's already impressive list of honors that include the Dixon Prize in Medicine and the Otto Warburg Prize. So it gives me great pleasure, and I ask you to join me in welcoming Susan Lindquist, who will be speaking on Alzheimer's disease, an entirely new point of attack.
Thank you. I have to say that it's really wonderful that now you've been really anticipating this talk, right? So you're really waiting to hear me. I'm going to talk not just about my work on Alzheimer's disease, but also about some work we've been doing on Parkinson's disease, because actually we're farther along on the Parkinson's disease. Alzheimer's is something quite new we've added to the mix, and I'll just be telling you about the beginning parts of the Alzheimer's disease. So, as human beings, we are incredibly attuned to diversity, and it's evolutionarily really important. We have learned to be able to recognize thousands of different faces, because in our history it has been really important for us to be able to recognize who is likely to help us find some food and who is likely to stab us in the back. So paying attention to diversity has been really important, but it has masked an underlying fundamental unity of living systems that has come to the fore recently as we've started to unravel and decode the genomes of different organisms. And that evolutionary commonality in history that organisms, very different organisms share, is something I'm now starting to take advantage of. So what this organism is here is a budding yeast cell. This is, many could say, mankind's best friend, responsible for all of the virtues of civilization, as the Romans defined it: beer, bread, and circuses. So circuses are molecular biology circuses. And in terms of being able to understand genetics and unravel complex biological problems that are simple, basic cellular problems, there is no organism on this earth that we understand as well as this one, or that we can manipulate as well as that one. So the fact that these organisms and these organisms have actually a lot of common features to them means a lot in terms of trying to unravel some of these very complicated diseases. So the theme of this day has been then and now, and so let me tell you a couple of aspects of that then and now. As I mentioned, genome sequencing of lots of different organisms has revealed how evolution has worked, and what that has meant is that we can realize that very complicated organisms evolved from simpler ones, and that profoundly enables discovery, because those simple organisms can be manipulated better than anything else, and they're cheap, and they're easy to grow, and they're fast. And so we can do things, when we can study something in those simple organisms, it allows us to move much more rapidly and inexpensively. Human genome sequencing has also been absolute revelation in terms of then and now. When I started out in science, we didn't actually have any idea how our genomes worked, the DNA code was working in us. We had some idea of how it was working in bacteria, but no idea of how it was working in us. And the sequencing of the human genome began at the Whitehead Institute, right here, in fact, in 1996. And the first complete genome, which cost more than $1 billion, and this was costs spread from all over the world, in fact, in many different institutes and places. But the conglomeration of all these different institutes, the largest part of it being done here, led to a single human genome sequence in 2001. And we're now sequencing human patients, and we can sequence the DNA of a human genome for a few $1,000s in a couple of months. So that's extraordinary enabling because we can compare the genomes of people who have disease with those who don't, especially when the diseases are genetically caused. That makes a big, big difference in trying to decipher what's happening. And the third thing I'm going to talk a little bit about, that all three of these things are kind of coming into my world of where I want to go, is stem cell technology. So we used to, the only thing we knew how to do was, and that was pretty amazing in and of itself, was to make stem cells, cells that can be differentiated into different body cell types, from embryos. Now we can make them from skin or from blood or from other tissues. And that means we can make them from an individual patient and study what's wrong with that particular individual. And we can use those potentially for personalized medicine at a whole new level that we had never really dreamt of when I joined this institute, quite honestly, just 10 years ago. And we can also use them for regenerative medicine. I'm using them more for the personalized medicine, but I think you'll appreciate it at the end, so how they might also be used in regenerative medicine. Although regenerative medicine, this institute takes a very broad view of that, and it includes both the personalized medicine use of stem cells and actual replacement of tissues. So common features of higher cells. So those yeast cells, believe it or not, as simple as they are, and we first started studying, the first people who started studying them were studying them because they wanted to make better beer. And they've been studying them for about 120 years now. And the thing about those yeast cells, they're simple single cells, but they're really very, very different from a bacterial cell. So bacterial cells are also simple cells, but they really started out a long time ago, a couple of billion years ago. But yeast cells and those types of higher organism cells came about on this planet about 1 billion years ago. So after 2 billion years of general life, then these eukaryotic organisms. Now, why am I saying eukaryotic? Eukaryotic just means a true nucleus, and that means there's a nucleus in here that contains all that DNA, all that genome I was mentioning. And cells that compartmentalize their genome and then compartmentalize all sorts of other important structures in the cell and divide it up into all sorts of different things. There's an odd humming that I wish we could get rid of if we could. Anyway, I'll leave that to the guys up there, see if they can help. All of this is a complex cell and the cells of our body. It's a schema of a complex eukaryotic cell. And it's the very same kind of drawing I would draw for a nerve cell or a liver cell or a yeast cell. The cells would be of different shapes, but they'd have these same things in them. So when we want to study what's going wrong in these complicated cells, the fact that these simple yeast cells and our brain cells have this similar architecture is very helpful. Now, DNA, the code of life, the genome, is a linear helical molecule. You've all heard of the double helix. And that's what's in that nucleus, that central region of the cell, command central sort of the cell. And to understand what that is doing in terms of coding for the complexity of life, I want to remind you of this cassette tape. And it's interesting, it's wonderful talking to an audience of this average age because you know what the heck this is. But this contains a long linear tape. It's a very simple looking thing. Doesn't look like it does much, but it has so much information on it. So, different parts of that tape specify different things, and quite complex different things, in the same way that different parts of our DNA specify complex proteins and complex functionalities in the body. So here we go. So the point really is that that's a linear piece of information, and it doesn't look like much. And DNA actually is a long linear piece of information too, but boy, it can encode some very complicated things. And different parts of it are used in that same way. Different parts of it can create a different kind of music by creating coding for a different type of protein. So what are proteins? Proteins are long strings of amino acids. And when they're first made, actually, they don't look very interesting either. And they don't do anything when they're first made. They're kind of a type of copy of the linear information on the DNA that represented the copy of linear information of amino acids. And they fold up, however, into these amazingly complicated shapes. And each protein, which is encoded by a different portion of this linear tape, each of those is doing something different. Some of them are muscles that are powering our movement. Some of them are actually getting energy. Some of them are digesting our food. Some of them are perceiving the light. They're really the basis of our thought processes. Proteins do just about everything in us. And so to give you a sense of how proteins, what kinds of different proteins are involved in this complex stew of making up that cell that I just showed you, I'm going to show you a movie, a very brief clip of a movie from an animation that was developed at Harvard by Ellen Veal and Robert Liu. Attempting, actually was done for the sake of undergraduates to try to give them a sense of what it looks like inside of a cell and how proteins go about their business and the different things that they do. I'm just going to show you a little bit of it. The very beginning of the movie has blood cells moving in the bloodstream. So you'll see these cells rocketing around. And then it zooms up to a much more highly magnified view of a cell rolling around another cell in the bloodstream. And then it goes much more high, very quickly, goes much more higher magnification. And you'll see the connections between two cells that are being made by very specific proteins. And then you'll see a bunch of other proteins skating around the cells. And I won't bother you with all the things they're doing, but just want to give you a sense of the dynamic and diverse nature of what's going on there with those proteins. So these are the blood cells. This is a cell moving along the artery. Here, those are proteins contacting each other, and they grab onto each other, and that's how these two cells are interacting. And these are membranes that separate the inside of the cell from the outside of the cell. They're lipid, that's oily substances. These are all different proteins that are moving around the cell. Many of the proteins are actually structural components. They help to give the cell integrity. Here's some more structural components. They're really quite beautiful, I have to, in my viewpoint. They are very dynamic. These are assembling. This is how these, you can have some idea of how a house is constructed. Well, we have some idea of how proteins assemble to form structures. Those structures get cut and reassembled, assembled and disassembled. Each of those individual things that's going around that's a different protein that's coming apart and coming together. Now this is really quite amazing. That's a protein that's moving a big blob of very important stuff from one end of the cell to the other. Oops. Yeah, so this is a protein, and it actually really does walk like that along another these are fibers made up of, as you just saw, hundreds of proteins assembled. And the cell wants to move. This is a lipid, a membrane-bounded vesicle that's an oily-like droplet that contains some interesting compounds inside. The cell wants to move it from one end of the cell to the other end of the cell. And this is very, very important in the nervous system, because these inside this vesicle are messages by which nerve cells communicate with each other. So that's an example of a protein. And this is actually anatomically correct. We actually have the crystal structure of these proteins. We know what their structure is at the atomic level. There's one thing only that's wrong with this movie. And it really matters a lot to my life. What's wrong with this movie is that they've taken out almost all the proteins so that you can actually see what's going on in there. Because if they had left all the proteins in it, you couldn't possibly see it. It's so packed So the problem I deal with when I talk about protein folding problems is this. We have to go from those very long amino acid chains to these very highly complicated assemblies. And the cell has to let those proteins fold in an environment like this. I mean, it's just crazy. It's so crowded. But it's not just crowded. As I was trying to show you with that movie, things are assembling and disassembling and moving around like crazy. So that if you take nothing else away from my talk today, take away the message provided by the following little film clips. When you think about proteins going about their business inside of a cell, swimming around in a cell, it's not like Esther Williams and her mermaids. It's much more like the wave pool at the Summerland Amusement Park in Tokyo on a really hot summer day. So that's really what it's like inside of a cell. It's incredibly crowded in there. And you can imagine then that if you were to put in some additional energy, and I actually first started studying this protein folding problem by looking at the effects that heat have on biological systems. If you were to put a little bit more energy into that wave pool, or if you were to have some of those, if you were to take out some of the water, or if some of the individuals in that wave pool weren't able to quite float right, you could get into disaster in a hurry. And that's the problem I study. I study the problems that are caused when proteins don't fold right. And it's a problem that you study all the time, every day of your lives, because you eat denatured proteins. You eat aggregated proteins. So these are proteins. That's protein. And this is what happens when those proteins get denatured and aggregated. Right? So that tidal wave pool I was showing you is that it doesn't take much to make these proteins aggregate into a mess, into a blob. And this is, whoops, whoa. That is one great big blob. But our cells have problems even when the blobs are much, much, much smaller, but in fact when they're microscopic. I think that this will still work, and if it doesn't, I'll just use my own two hands. Yeah, it does. Okay, so these are slices from human brains, people who have died of various neurodegenerative diseases. And actually, this protein folding problem has taken me into all sorts of different realms of biology. I was originally really just doing basic science, but it's taking me into studying cancer biology and infectious diseases and many things, because it's such a fundamental, basic problem. But the thing that I'm really the most excited about is this study of neurodegenerative diseases. Now, these look kind of nondescript and uninteresting to you, but let me just tell you what is common about these. There are these blobs in here that are made up of aggregated, misfolded proteins that are rather like that egg white that has changed to a hard mass. And these aggregated proteins are different proteins in different diseases. And in Alzheimer's disease, you may have heard of A-beta. You may have heard of plaques and tangles. That's what those plaques and tangles are made up of, is badly folded proteins. And in Parkinson's disease, it's a protein called alpha-synuclein. Now, in terms of this, the then and now theme of this day, unfortunately, both then and now, there's not a single treatment aimed at correcting the underlying cellular pathology in these diseases. Not one. There are palliative treatments. There are things trying to help fix problems once they've occurred, but nothing aimed at trying to stop that underlying fundamental beginning cause of the disease. And so you might ask, why? And the reason is because it's so difficult. And why is it so difficult? It's not that people haven't been passionate about trying to find things that would work on these diseases. They have been. It's not that they haven't been working hard. They have been. But it's so darn difficult because these diseases are very complex. They're not like sickle cell anemia, which is caused by the misfolding of just one protein, or many other diseases which are caused by the misfolding of just one protein. And even for those diseases, we don't yet have cures. There's just a beginning now, is starting of having the first treatment for a protein misfolding disease that's actually directed at the individual misfolding protein is just about ready to be approved. But then these diseases are not just one protein, major protein is misfolding, but there's all kinds of reasons for it. There's all kinds of other things that cause it. So it's a really very complicated problem. And until we had these three different things going for us, the fact that evolution happened, and in fact you can use simple cells to study complex problems because there's fundamental commonalities between them. The fact that the genome sequencing has allowed us access to finding what's wrong in certain people that predisposes them to certain diseases. And the fact that stem cell biology has just exploded means that we can put those things together to create new discoveries and try to find new cures. So I got the idea a while ago that maybe, in fact it was about 10 years ago now, that maybe we could take some of these proteins that are misfolding in the brains of people with these horrible diseases and put them into those simple cells, those yeast cells, and see if they would also misbehave in those cells. And if they did misbehave, if we could figure out how to stop it from happening. And the general reaction to that was this. Come on, you know. How can you possibly conceivably study a neurodegenerative disease, which is such a complicated thing about brain cells which are trying to communicate with each other when you're in a single cell that's not communicating with other cells, it's just on its own. But if you want to study something that's really basic and fundamental, it's actually, not only is it that with yeast cells we have a system that we can manipulate better and manipulate more rapidly, we also have single cells, so we don't have the complexity of secondary and tertiary and other things going on in terms of the communications between cells, at least not initially. We need to focus on the basic initial precipitating cause. Having a single cell, cells that normally grow as single cells, just makes the problem actually a little bit less complex. So I'm going to tell you about some progress we've made with Parkinson's disease first, and as I mentioned, I'll mention Alzheimer's disease. But Parkinson's disease, that blob that's present in those nerve cells of patients with the disease, is called alpha-synuclein. And the thing is, it's a very small protein, so it's a simple protein. That was one of the reasons why it was one of the ones we started with. Very simple little protein. And it doesn't fold up very well unless it's associated with membranes, those lipid-bounded vesicles that I told you were moving around the cell, the nerve cell, trying to get from one side of the cell to the other. When it binds to those types of membranes, and it only gets structured when it does. When it's not bound to those, it can form all kinds of misfolded species that are really bad actors in the cell. And we figured, gee, these are pretty simple aspects of the biology of this protein. Maybe we really could study that in a simple yeast cell. And it's turned out that it works. So to give you a little background on Parkinson's disease, it's the second most common neurodegenerative disorder after Alzheimer's. It's generally characterized by the loss of a particular type of neuron called the dopaminergic neuron, and that's what gives from a particular region of the brain, that's what gives the particular symptoms, is that those particular brain cells, neurons are brain cells, are affected first and most severely. And there are a bewildering number of environmental factors that seem to be associated with disease. One of them is manganese exposure. Another one is certain types of toxins in the environment. There are many genetic factors. It's clear that there's multiple genes that can lead to this disease. But what's really cool is that not only do you generally see one particular protein in those blobs that identifies that as being something central to the disease, but we also know from the genetics, from DNA sequencing, that simply having too many copies or having a mutated copy that it doesn't fold right, but even just having too many copies of that protein gene can cause disaster. So that was very enabling for the thought that we might be able to create simple models in our yeast cells by simply expressing that protein in the yeast cell, and then maybe expressing a little more, and seeing what happened. Another thing that was helpful to us was knowing that that protein is a bad actor not just in Parkinson's disease, but also in something called dementia with Lewy bodies and something called multiple system atrophy. So although for a while all the concentration was on particular individual types of nerve cells that were being affected in Parkinson's disease, we were then beginning to understand that, wait, this protein can go wrong in lots of different cell types. So we thought, well, maybe it could also go wrong in a yeast cell. And so we made a simple model. We took that protein, and we attached this kind of magical molecule called GFP, or green fluorescent protein, to it. What that is it's a molecule that actually was taken from jellyfish. And you may have experienced jellyfish out in the ocean that fluoresce green. And you can stitch up your protein to that protein, and they'll make one bigger protein. And you can actually look at the cells under the microscope and see what's happening to your protein, because it shines green light at you. So we put the protein into yeast cells, and 2 copies, rather kind of mimicking the 2 copies that are in human cells. And it went to the outside periphery of the cell. It went to the membrane, and that was great, because in the human disease, it's known that it's at the, when I was showing you those big bowling balls, they were moving along, those are membrane bounded, and they're moving to the end of the cell. They're moving to the place where they're going to transmit from one cell to the other, these chemical signals. So it was going to the same equivalent place in a yeast cell and a nerve cell. And when we expressed just a little bit more, disaster happened. And these cells grew normally, those cells grew slowly, and those cells died. So this is quite unusual. There are many proteins that when you make too much of them, the cells will get sick. But not very many where it's such a small difference in amount can go from being fine to being a disaster. And yet we knew that that was true not only in humans, but in yeast. So now we went to a library. We went to a library of DNA sequences. So just like I told you, we had this section of the tape that can code for the Charleston, or that can code for Twist and Shout, or that can code for a Beethoven piece of music. We have individual pieces of the DNA of the genome of this organism. Every single little section of that DNA has been put into what we call a vector that we can move around into other cells, into our yeast cells. And so we could ask for every, you can do it in this organism, you can't do it in any other organism with anything like this speed. We could ask then, if this library, we would grow our yeast cells in each well of a culture dish, and then each cell had a different additional gene growing in it. And we asked for what things would make these cells better and what things would make these cells worse. And we took those, we had these cells growing in 96 well plates, and then we stamped them from one plate from that plate onto an agar surface, and this is what that looks like. You can see all these colonies are not growing very well, but this one here has a gene in it that makes it grow better. And this one here has a gene in it that makes the cells worse. So we were able to screen through every piece of DNA in the entire yeast genome for things that made things better or worse. And we got out 60 things that mattered. That's a lot. And this is, again, an example of the complexity of this disease. But here's another little mini then and now. Robots. So we used to stamp our plates like that. In just the last 2 years, we've got robots that allow us to stamp them in replicate and quadruplicate so that it's much more reproducible and much, much, much faster. And that's just happened in the last 2 years. Anyway, so we got out these genes from the yeast, and they coded for certain proteins, and we knew that those proteins were making the yeast cells better or worse, but of course, no one believed us that they could possibly be important. Until we put them into nerve cells, and we used nerve cells from a variety of different organisms. We used nerve cells from a nematode because they're actually hardwired up in the nematode in a see-through worm. It's one of the most amazing organisms on the planet. You can see through the surface of this worm. You actually see the nerve cells and what's going wrong with them, and you can actually test them in the same type of the dopaminergic neurons that go wrong in Parkinson's disease. We also did that in fruit flies. in collaboration with these other laboratories. And we did that in primary neuronal cultures, that is brain cells from a rat brain. And we're now doing that with Rudolf Janisch's group with stem cells from human patients. So we take these brain cells from the rat, which is where we're more and more frequently going directly from the yeast cells to the rat cells. And we take those brain cells and we disrupt them in culture and we plate them, and that takes 5 days. And then we have viruses that will now have a library of these coding sequences for the human genome. And we add those viruses to those brain cells, let them grow for a while, and see if they also make them better or worse. And lo and behold, of the 13 things we got out of a yeast cell that made the cells better or worse that we tested so far in this system, 12 of those 13 work in the nerve cells. Here's another one that validates this approach, this crazy approach. DNA sequencing showed us that there was a very similar gene. One of our genes had a very similar homologue in humans. In fact, of those 60 genes that I told you about, most of them had it was very clear that humans were making that same protein. But we didn't know anything about this gene, even in a yeast cell, until about 6 months after we identified it. This paper came up with hereditary Parkinsonism with dementia is caused by mutations in this gene, which does that. Actually, it's not much to know about it. It doesn't tell you very much about it. But we realized, oh my God, we've got this gene from yeast cells, and nobody knew what it was doing. But it turns out that when it's not working right in a human, it causes Parkinson's disease. So that was another validation of the fact that the things we're seeing in this yeast cell matter in human biology and medicine. And we were then able to use our yeast cells to figure out what that protein was doing. And what that protein was doing was detoxifying manganese. And that was important because manganese exposure has many, many different connections to Parkinson's disease, from mining to urban pollution to welding, to actually drug abusers whose recreational drugs get contaminated with manganese. So finally, and I'll get through the rest of the story pretty quickly, we did a chemical library screen. So there are also libraries of chemical compounds, and Harvard has a library of 150,000 compounds that we went to. 150,000 compounds. Now, can you imagine testing 150,000 compounds in, say, a mouse? Or even in rat cells in culture, the expense and difficulty of that? We were able to test 150,000 compounds. We simply looked for things that would make the cells better. And we got a small number of them. And the reason why we embarked on this in the first place, you might think, why would you look for compounds in a yeast cell that couldn't possibly help a human being? Well, this is a drug that I would venture to say that probably half of the people in this room are taking, and I'm certainly taking it. Lipitor, simvastatin, that works just as well in a yeast cell, as do many of these other blockbuster drugs. And the reason why they work both in a human cell and a yeast cell is because they're hitting the same target, because these cells are so similar. So we found then, when we took those compounds and asked whether they would fix the nerve cells in the nematode, they did. Oops, this is the AD model, sorry. I got these a little out of order. Anyway, they not only fixed the problem in the nematode, they fixed the problem in rat midbrain cultures. They took those bowling ball aggregates of protein and made the protein go back to the membrane and behave itself in a yeast cell. And then we took another toxin that's associated with Parkinson's disease, but it wasn't known whether it was associated with this alpha-synuclein protein at all. We took another toxin, exposed neurons to that toxin, and asked whether our compound that we got out of yeast cells would fix the nerve cells from that toxin. And it worked. So this allows us now to say that what's going wrong for that particular protein is a common thing that goes wrong with lots of every eukaryotic cell. It would go wrong if you were making too much alpha-synuclein and you weren't doing the right thing with it. It just means that nerve cells, that particular type of nerve cell is more sensitive when that goes wrong. And the reason is because they're really dependent upon that trafficking, moving those big blobs of neurotransmitters, neurosignaling molecules around from one end of the cell to another. A nerve, if that goes wrong in a yeast cell, they can cope a bit, but a nerve cell can't. And all these other different things I won't go through, but basically what it is just the nerve cells are dying, those particular nerve cells are dying first simply because they're more sensitive to that same fundamental underlying problem. Now, what about Alzheimer's disease? Well, this is the most common and devastating neurodegenerative disease. We've done a library screen in yeast. Again, we screened 5,000 genes. We screened for things that would correct growth. We got genes that had clear human matches. We found that several of them worked in the worm model, and 2 protected cortical neurons from A beta, that protein that forms plaque in the brains of people with A beta. And 3 of them have recently been identified as being risk factors for Alzheimer's disease in people. When they are broken, Alzheimer's disease is altered in people. So again, this tells us that our simple yeast model is providing us with some insights. Now, I don't want to begin to tell you that we're going to be able to study everything in the yeast cell. There's whole realms of the biology of the nervous system we won't be able to study. But again, getting at this fundamental underlying problem, we think we've got a handle on it. So here's an example of nerve cells in the tissue culture dish. Nerve cells with that bad actor A beta protein put on them, and it kills them. And here's one of our genetic factors that rescues, that's an Alzheimer's disease risk factor. Again, we've done a chemical library screen. Now, what's really amazing, especially since we've only started this work, is that we already have a compound that works in a mouse Alzheimer's disease model. Not only that, that compound rescues that mouse not only in terms of restoring pathology, it also restores cognition. And the reason we were able to do that so quickly is because that compound was already being worked on slowly, painstakingly over the years in the laboratory of Audrey Bush and Rudolf Tansy. It's actually now in clinical trials for humans, and maybe it's way too early to tell whether it's going to work. It could have all sorts of complications. But the fact that we were able to screen 100,000 compounds in yeast, we only got out maybe 50 that hadn't made any difference, and that was one of them. Gives me hope that, again, we'll be able to find things that will, using this simple system, that will matter. So where do we want to go? Then, now, future. So we now have patients that are all given the same diagnosis of Alzheimer's or Parkinson's or all these other dreadful neurodegenerative disease. It's the same diagnosis, and they get the same prescriptions, some of which work, some of which don't. They're really just treating symptoms. What we want to do is to be able to find which individual causes are responsible for these individual diseases in these people. And we're doing that in collaboration with 2 people here at the Whitehead, Rudolf Janish's laboratory and Janif Ehrlich's laboratory. We're taking skin cells from patients, and we're deprogramming those patient cells into stem cells. Those differentiated skin cells go back into a very simple early fate. Then we take those early fate cells and we redifferentiate them into nerve cells. And the reason why we would do all this complicated mess is that we'd like to see what will make the nerve cells of Parkinson's disease patients behave better, more like the nerve cells of normal patients. And we've got some challenges here, some major challenges. Here's one. These diseases develop in patients in late adult years. How do we replicate something that takes 60 years to happen in a human being in the tissue culture? Well, what we've found is that we're beginning to find, and we really think that this is going to work, that we can apply different stresses to those cells that will cause them to undergo that type of aging process more rapidly, and that we'll be able to, in fact, study these things. And some of the things that we can do, we know we can do, are to use those toxins and manganese exposure, things like this, which we know are relevant to the human disease, as well as our genetic factors. So what I'm hoping is that we're at the tip of the iceberg here. I really hope this for all of our sakes, because I'm getting into that age now where this is something that's facing me as well. But this is just really one type of an approach, but I hope it will give you some excitement that diseases that used to seem to be absolutely, completely no way, no how are we going to be able to do anything about it, I think will be attackable. And I want to thank you. I want to thank you for listening, and I also want to thank you for your support of the Whitehead. It means so much. I spend an awful lot of my time writing grant proposals, as do the other members of this institute. And quite often, those grant proposals, especially if you're trying to do out-of-the-box, really bold, unusual things, like study a neurodegenerative disease in a yeast cell. Let me tell you, I could not get support for that from the NIH. And I was able to get some support from the Howard Hughes Medical Institute and some support from the Whitehead to get this thing started. And that's why this place is just such a really amazingly wonderful place. Makes a difference in this world. And the final thing I want to do is just thank the amazing people who've been involved in this. I'm not going to name them specifically. I'm just going to show you their faces, and I hope that what you'll get out of this, my showing you these faces, is the sense of excitement and passion and intelligence and verve and vigor that these wonderful, amazing young people bring to this. And this is my wonderful collaborator, Rudolf Janisch. This is finally dying on me, and those are the folks in his lab that we're working with. Janne Verlich is just starting here at the Whitehead Institute, and we only have, we're collaborating. Oh, sorry, that's what should be Esposito. And I think the spell corrector corrected that. Anyway, and these are my wonderful collaborators. Actually, the world of doing science has changed so much lately, because if you want to really move fast, what you want to do is reach out to people all over the world who've got something that you don't know how to do, tell you how to do something, and I'm good at that. I've been putting together some really cool collaborators, and I've had the most wonderful, wonderful group to work with. So that's it, folks. Yeah, so we started actually 15 minutes late, and I actually finished in 5 minutes less time. So I hope there's time for questions. Yeah.
If you can speed age up, then a disease like progeria, the advancing of age, or a better question is, can you slow age down and find the fountain of youth?
So progeria is a really tough disease. We actually know some of the mutations that are responsible for progeria. The question was, if we can age things, can we slow down the process? Like progeria is a syndrome, a horrific syndrome. Thank God it's very rare. But it causes very rapid aging. And even 15-year-old kids actually look like they're 90 years old. It's a terrible disease. And we know the fundamental underlying cause of that. And it's going to be hard to, I'm not saying it's going to be impossible to fix it. It's going to be hard to fix it. But at least we know the underlying cause for several of the different manifestations of that disease. I'm not working on that one. There are some other wonderful people who are working on it. But can we find the fountain of youth? Well, that, it used to sound like pie-in-the-sky craziness. But I do think that different people age at different rates. And that's in part due to our genetics. And so there are people out there, and there's an individual at Mount Sinai that we're going to be collaborating with, actually, who's studying people who live into their 100s in very healthy lifestyle. And then and trying to find the DNA mutation, the changes in the DNA that matter to that healthy lifestyle. What's going to be helpful for us is that we can then compare those changes with the kinds of changes that we're seeing in these early onset diseases when we're sequencing genomes. So we've just been funded, at least part of the study has been funded, to sequence the genomes of about 2,000 patients with Alzheimer's disease and 2,000 patients with Parkinson's disease, and 2,000 controls. But that one study will actually provide an independent set of controls. And so I do think there's two aspects of this. What are the overall aspects of aging that matter? And there are many, many people working on that. I'm not working on the overall aging problem. But I think we're going to find things that matter. That, in other words, I don't think we're going to be able to push the human lifespan beyond 150 years. But I wouldn't be at all surprised if the average person could expect, you know, in the not too distant future, to be living a longer, healthier life because of new discoveries. What I'm working on is the particular interface between the aging process and individual things that are going wrong and making, before someone should be really experiencing the miseries of old age, is causing people to have these earlier onset diseases, these diseases that strike them in their 50s and 60s and 70s, and really ruin their lifestyle, really change things profoundly. So I'm working more on those particular things than the general aging process. Yeah.
Susan, just I think last week I read that the plaque that it was thought to be the cause is actually a protection from. So how do you?
Yeah. So that's great. It's wonderful to hear you guys are reading like that, and it's such a lovely question to answer because, believe it or not, we had a very early hint that that was the case in yeast cells. We have been working on something called yeast prions, and there are prion diseases you may have heard about called mad cow disease, which is a protein misfolding disease. And so we've been studying, yeast have their own prions, and we've been studying those proteins. And we found that what happens is that when the protein is soluble and individual and just by itself, it's not harmful. When it's assembled into these big, large units that are like the plaques, big, long assemblies, they're not harmful either. It's when they first start to assemble into these, they're flickering around and they're conformationally flexible. They're misfolded, but because they have a lot of surface area, they can when they're in their small little pieces of misfolding, they interact with other things and they cause all kinds of bad things to happen. We see them in the brain of patients who've died of these diseases as big aggregates because things have really just gone a long way, right? But it's the earlier forms, the smaller misfolded forms that are called oligomeric species that are likely to be the bad actors, and that seems to be true both in our yeast cells and in the nerve cells. So it's a very interesting aspect of this, but it's not in any way actually counter to what I've been saying. And I'm so glad you asked the question because we had this result in yeast cells about 6-7 years ago that if you could actually, what you really don't want to do is dwell in these flexible, interactive, misfolded little things. You could either, you really want to return the protein to a normal state, that would be the best. But if you could get it to assemble in an ordered array so that it wasn't touching so many different things, that would be a little bit better too. So, yes?
You talked about aging cells more rapidly with stress. And I'm just wondering if you could comment on the physiology of stress for a minute, you know, whether it's road rage, pain, road rage, or angry boss. Stressors, in fact, age us, and do they accelerate neurodegenerative diseases?
Yeah. So there's no question that aging, protein folding is a very, very important proponent of aging. Psychological stress, does that happen because it's raising our blood pressure and raising our temperature? A lot more to it than that, of course. But there's a creature called the naked mole rat, which is my nomination for the ugliest creature on the planet, and you can look it up on the internet tonight if you would like to take a look at this. It's called the naked mole rat. It's rats and rodents of all types live a couple of years. This naked mole rat lives to be 35 years old. Okay? And when you go in and look at its proteins and their propensity towards misfolding, in a time when those proteins should be all misfolded, they're not. And so this aging process and trying to figure out, well, what is different about the genome of that naked mole rat? It's really funny because there are these colonies, there are a couple of different colonies in the United States, and they're relatively small. And now everybody wants to get their hands on these naked mole rats. And of course, you want to get the 35-year-old naked mole rats, right? And so there aren't that many of them to go around. So it's a really an interesting situation. But it's very clear that the metabolic syndrome that leads to more rapid aging also leads to more rapid protein misfolding. And that's one component of it only, but it's one component of it. Yes. Yes?
I wanted to ask you about your process for using yeast to screen all these genes out. And what I'm thinking along the lines is, what if it's maybe a little misleading because the yeast is so much simpler than a mammal cell, and would you be eliminating compounds that didn't work in yeast, but that might have worked in a mammal?
Sure. So the question is, would we be eliminating compounds that, by using a yeast screen, would be missing things? Just because it wouldn't work in a yeast cell, would not work in a yeast cell, but it might work in a mammal. Of course. We're going to miss all kinds of things in yeast cells. But the fact that we can get some, let me tell you, we can screen through 150,000 compounds in the time it would take to screen through 4 or 5 if you were using higher organism. And we have just been funded by the NIH, National Institutes of Health, which didn't want to have anything to do with this work for quite a while. They've just funded us to do 3 screens of 350,000 compounds, one for our alpha-synuclein Parkinson's model, one for the A-beta Alzheimer's model, and another one for an ALS model. So they're beginning to drink the Kool-Aid too, so I don't know. But the thing is, of course, we're going to miss all kinds of, there'll be whole realms of the biology we won't be able to touch in yeast. But if we can use the yeast cells to quickly identify some things, boy, we're going to be ahead of the game. And moreover, there's nothing to prevent us from taking other proteins that as we do our genetics and try to find things that have homologs, that have clear related proteins to the human disease, and putting those proteins into a yeast cell. Or especially what we're going to go after first are the ones that are already similar to the yeast proteins. Yeast cells already have proteins that are related, and a lot of them are, 50% of them. Yeah.
Have you identified any protein misfolding associated with plain old garden variety dementia that is not Alzheimer or Parkinson's or ALS related?
Not schizophrenia. We haven't touched schizophrenia yet. Oh, I'm sorry. Dementia. Oh, dementia.
What's happened to me?
Plain old dementia of aging. Senile dementia. What is senile dementia is not entirely clear. Is it just Alzheimer's disease or Parkinson's disease that hasn't been diagnosed properly yet, or is it some... What really, each of those diseases is not a disease. Each of those diseases, like you've heard, of course, that cancers are many different kinds of cancers. Even Parkinson's disease, although there's a commonality in that they almost all people with that disease, who a neurologist would say, you have Parkinson's disease. Almost all of those people have this misfolded particular protein. But they have lots of other genetic factors contributing. Quite a few people have both A-beta, the Alzheimer's disease thing, and the alpha-synuclein protein, both of them, but at lower levels, and the combination of the two matters. But we're really only just beginning to find out what are the causes of what we would call idiopathic senile dementia. But, and I'm not going to tell you that protein misfolding is going to be at the basis of all of it, but I think it will probably have an important place.
Part of the reason for the question is that I had the opportunity to meet the developer of Azilect, which is for Alzheimer's, right? And Parkinson's, partly. But
partly, yeah.
But he's saying, as far as he can tell now, he's not sure what comes first, the dementia.
Exactly.
Which is
no, it's a spectrum. It's what they call a spectrum of disorder. Each of those diseases is a spectrum of disorders, and different people will have a different spectrum of symptoms. And The reasons for that is that they have different combinations of underlying causes, and they're not completely unrelated to each other. They're partially overlapping, and they're going to be partially overlapping with other causes of disease. However, what's happening with these genome sequencing efforts is we're going to at least be able to get at several of the different underlying genetic causes, because as sequencing gets cheaper, and as we take more and more patients and study more and more people who have non-differentiated idiopathic senile dementia versus the ones that don't, as we start to get now not a couple thousand, but 100,000 of those individuals, I think we'll be able to identify some of the underlying causes for these and the commonalities. But again, I don't want to hold out the false promise that we're just on the verge of cures for these diseases. There's going to be some of them are going to be much more complex than others. And as I mentioned, we still have some very major hurdles in terms of we're now getting these stem cells and we're differentiating them into nerve cells in the culture. We've got to find out an assay that will be really robust and show us the difference between the patient nerve cells and the normal nerve cells. And that's one of our major challenges right now. But I'm sure we're going to do it. I think we'll do it. So I don't know who to pick. Maybe David could, yes? Yeah. A lot of wonderful questions. I'm sorry. I wish I could answer them all. Yes? Similarities in other cultures. Well, what's really interesting about this is that these diseases are overall found in all cultures. So all cultures have Alzheimer's disease that I know of. There are certain neurodegenerative diseases and certain causes, for example, of Parkinson's. Certain causes of them are more common in certain populations than others because of the inbreeding, and those mutations are more common in that population. But one of the reasons why these neurodegenerative diseases are probably around is that the things that cause them were not selected against in evolution, because human beings never lived that long, right? You were lucky if you got to be 30, 32, And during all the time when evolution was operating on us and getting rid of the genes that mattered for our survival and for our being able to leave progeny behind, like leave children behind. And things that caused or didn't cause Alzheimer's disease just were not part of that equation. So it's really a very common Chinese and Aborigines in every population you see those diseases.
Take one or
Yep.
two more questions.
What is the incidence of these diseases in blue zone people?
In what?
The blue zone people.
What is the incidence of these diseases in the blue zone? The what?
Blue zone.
I don't know what you mean. I'm sorry.
You're going to have to.
What is it in blue zone, you said, but I don't know what blue zone is. I'm sorry.
Somebody's written a book about the places around the world where there are superannuated people, older parents going around and straight. It's not the kind of research you do here.
Yeah.
Sociological research. But I just wondered, because there's greater populations of these people who've reached great old age. In those populations, what has been the incidence of these diseases?
Well, I would say by definition, if you reach great old age, you're not getting Alzheimer's disease. But as I said, there are, I don't know anything really about this blue zone thing, I'm afraid. There are certainly centenarians, and there are certainly centenarians where it runs in the family, where it's genetic. We had a lecture here at the Whitehead not too long ago about a guy who was studying what they call centenarians. To be a centenarian and to be in the study, you have to not only be 100 years old or better, without dementia, without any serious illnesses, without diabetes. You have to have a first-degree relative who's also like that. And there was a family that had 5, where the youngest one just died and she was 102, and they couldn't figure out why she died so young. I mean, really. So there are a lot of genetic factors that play into these diseases.
Take a question up here.
Yeah. So these compounds that you found that basically reverse function, presumably allow proper folding. Do we know anything about the pharmacology, about chemistry, from a structural biology standpoint, that might into the same domain?
Very, very good question. Do we know anything about the pharmacology of these proteins and whether they're targets? And we haven't yet got the target of any one of them. The one that I mentioned, that I actually founded a company several years ago that worked on an orphan disease. It's a very rare disease, and there's a small number of people who get this neurodegenerative disease. When they do, they will invariably progress rather rapidly. And so we were able to find a compound that actually, in that particular case, we were able to find a compound that bound to that protein and prevented that protein from misfolding. And that, I think, is going to be approved very, very soon. And I learned so much in that process. It was just an amazing thing. But I no longer am associated with that company. It's gone on, and it has its own life. It was bought by Pfizer, actually. But the other compounds that we've been looking at so far, they don't seem to affect the folding of that individual protein that's misfolding. They don't directly bind that protein, but they're affecting some other things that are in the equation, and we don't know quite what it is yet. It's a level of discovery I really want to get into in a big way, but we're just starting that. But I showed you that we had those. I was showing you the big bowling balls of protein because it's easy to see. But the same cells that have those big bowling balls actually have lots of these other little ones. And the little ones are the thing that's the real problem. But we had some compounds that pushed that protein back to the membrane, right? And I showed you the things had gone bad, and then in another cell type they had not gone so bad. It was back in the membrane. But we know it's not directly binding that protein. It's doing something else. And that fits with these other things that we've seen, because the one case I told you about the manganese poisoning, manganese isn't binding that protein either. Manganese isn't directly binding that protein, causing it to misfold. It's affecting other processes in the cell that then lead to problems that then cause that protein to misfold. So it's complicated.
Let's take one. We got lots of questions. Let's take one last question. We'll save the rest for the break. Yeah.
Do you believe that brain exercise, like attending this colloquium, stave off these diseases?
Yes. Without question.